Today, we are diving into one of the most incredible feats in all of nature. It's a story about speed, about mind-bending precision, and about how every single one of your cells manages to pull off a truly impossible task. It all boils down to one single question.
I mean, really think about that for a second. Every time a cell divides, which is happening in your body right now, it has to duplicate its entire genetic code, this gigantic, sprawling instruction manual, and it has to do it almost perfectly. How on earth is that even possible? The scale of this is it's hard to even wrap your head around.
3 billion. Just let that number sink in. That's how many letters, or what we call DNA base pairs, are in the human genome. It's an entire library of information, and it's packed into a nucleus so tiny you could never see it. And your body has to copy every single letter. But you know, the real challenge isn't just the size of the book. It's the deadline. Your cells have to make a perfect copy of that entire 3 billion letter manual in roughly 8 hours. So right away, we have this massive biological puzzle. How do you copy something so huge, so accurately, and so incredibly fast?
So here's where things get really wild. You'd think that to solve this speed problem, our cells would have some super fast copying machine, right? Well, nope. Our cellular machinery is actually about 10 times slower than a simple bacterium's, and our genome is 1,000 times larger. Logically, this should take forever. So how do our cells do it? Well, they use a classic strategy: divide and conquer. Instead of starting at one end of the book and reading to the other, our cells open the book to tens of thousands of places at once and start copying everything in parallel. It's like having 100,000 workers on the same project all at the same time.
To pull off this incredible job, the cell uses a whole toolkit of specialized molecular machines. These are enzymes, each one designed to solve a very specific problem that comes up during replication. So, let's meet the crew.
First up, you've got helicase. Its job is simple, but absolutely essential. Think of it as the unzipper. It latches onto the DNA double helix and just plows forward, unwinding the two strands so that the copying machinery can actually read the code that's hidden inside. Without this guy, nothing else can even get started.
Okay, next we have the star of the show, DNA polymerase. This is the master builder. It's the enzyme that actually reads the template and synthesizes the new strand of DNA. But, and this is a big but, it's a bit of a diva. It has two very specific rules. It can't just start building from scratch, and crucially, it can only build in one direction. It's a one-way street, and that one little rule creates a really fascinating problem for the cell.
So how does a cell deal with this one-way street problem? Well, it gets clever. For one of the strands, the leading strand, it's easy. Polymerase just cruises along, building one long continuous piece. But the other strand, the lagging strand, is facing the wrong way. So the cell solution is to copy it backwards, in a bunch of small, separate chunks. Then another enzyme, a real hero called DNA ligase, comes in behind it. Its job is to act like molecular glue, stitching all those little fragments together into one perfect, complete strand. It's such an elegant solution.
But hold on, all this unzipping creates yet another huge problem. I want you to imagine trying to untwist a tightly coiled rope by pulling it apart in the middle. What happens? The ends just get wound up even tighter, right? The exact same thing happens to our DNA. All that unwinding creates this incredible tension ahead of the replication fork, and if it's not dealt with, it would literally stop the entire process cold.
So how do you solve that? Enter an enzyme with a jaw-droppingly cool name, topoisomerase. This thing is the ultimate detangler. It speeds along ahead of the helicase, finds these super tight coils, and what does it do? It makes a tiny, controlled cut in the DNA backbone, lets it spin around to release the tension, and then instantly seals it back up. It's an amazing little piece of engineering that prevents the whole assembly line from getting into a massive traffic jam.
Okay, so we've solved for speed, we've solved for direction, and we've solved for tension. You'd think we're home free, right? Well, not quite. For creatures like us with our long, linear chromosomes, this whole process creates one last major headache. What happens when you get to the very, very end of the line?
And honestly, this is the perfect analogy. Think about the little plastic tips on your shoelaces. They're called aglets.
They don't actually help you tie your shoe, but what they do is crucial. They stop the ends from unraveling and fraying. Well, it turns out our chromosomes need the exact same kind of protection for their ends.
Here's why this is a problem. Remember that lagging strand, the one that's built in little pieces? Well, when the replication machinery gets to the very end of the chromosome, there's no place for it to lay down the final primer to start that very last chunk. So the result is that with every single time a cell divides, a tiny piece of DNA is lost from the end of the chromosome. You can see how, over time, that could become catastrophic, eating away at important genes.
So the cell's solution to this is it's brilliant. It's these things called telomeres. They are quite literally the biological version of those plastic shoelace tips. They're these long, repetitive sequences of what is essentially junk DNA that are tacked on to the ends of the chromosomes. They serve as a disposable buffer. The cell can afford to lose a little bit of this protective cap with each division because no valuable genetic information is being lost.
And what's so incredible about this is that the shortening of these telomeres acts as a kind of cellular clock. A brand new cell starts out with nice, long telomeres. With every division, they get a little bit shorter and a little bit shorter. After about 50 divisions or so, those telomeres become critically short, and that sends a signal to the cell, telling it, hey, you're getting old, it's time to stop dividing. This is a fundamental part of how our cells age.
All right, so the cell has this incredible system for copying everything. But what about mistakes? What about typos? This whole process isn't just about being fast, it has to be almost perfectly accurate. So the cell has an entire quality control department that is constantly on patrol, looking for and fixing errors. The accuracy is just staggering. DNA polymerase, the master builder, actually has its own built-in proofreading function. It catches most mistakes the second they're made. This brings the final error rate down to just one mistake for every billion letters copied. That's incredible. But think about it. With a 3 billion letter genome, that still means a few typos are going to sneak through every single time a cell divides. And that doesn't even count damage from things like UV radiation.
So to handle all this, the cell has a whole crew of different repair systems on call 24/7. You've got mismatch repair, which basically acts like a spell checker, zipping along the new DNA to find and fix those single-letter typos. Then there's excision repair, which is more like a patch-up crew. It can cut out bigger chunks of damaged DNA, say from a sunburn, and replace it with a fresh patch. And for total catastrophes, like when the DNA strand breaks completely in two, you've got this emergency system that acts like welders, doing its best to fuse the ends back together.
Okay, so we have all these amazing tiny machines for replication, for repair, for managing aging, but they don't just run wild on their own. They're all part of a much bigger, tightly controlled network that literally governs the life and death of every single cell.
This whole operation is managed by a series of what are called cell cycle checkpoints. The best way to think of them is like mission control before a rocket launch. Before a cell is allowed to copy its DNA, or before it's allowed to divide into two new cells, it has to pass a whole series of strict inspections. These checkpoints scan for DNA damage, they make sure all the resources are in place, and they only give the go signal when everything is absolutely perfect.
So let's just step back and look at the big picture here. You have DNA replication, making the copy. You have the repair systems constantly proofreading and fixing that copy. And you have the cell cycle checkpoints acting as the master supervisors of the entire process. If the damage to the DNA is just too severe to fix, the cell does the responsible thing. It triggers a self-destruct sequence called apoptosis. It's when this beautiful, delicate balance fails, when the checkpoints break down and cell division just runs out of control, that you get diseases like cancer. It's a system where control is absolutely everything.
And that leaves us with this one last, really profound thought. We've just gone through this astonishingly complex system that is built for near-perfect accuracy, because mistakes in our DNA can be fatal. And yet, on the grand scale of life, it's those very same mistakes, random mutations that create diversity and drive all of evolution forward. It really makes you wonder, in the grand blueprint of life, what is the perfect rate of error? What's the perfect balance between stability and the change that allows life to adapt and thrive?